Hi everyone. I just wanted to recap what we're doing this week and also talk a few minutes about the most difficult material on Unit 1. And of course, please reach out to me with questions. So this week we're wrapping up Unit 1. And so what we've been working on in the notes is the nucleus, the structure of the nucleus, the structure of DNA, the structure of RNA, and then basically how DNA works. And I put a question in the discussion, which I appreciate people, talking with me there about DNA. And I just wanted to say a few words about the notes, maybe help you out a little bit, hopefully. And of course, all this material is already there on Brightspace, but, just in case this helps. What you want to think of when you think of the nucleus and DNA is the fact that nucleic acids, there are two different types of nucleic acids. Nucleic acids are just, at one point, the acidic material within the nucleus. We didn't even know about the existence of DNA and RNA. And so it was later discovered that there are two types of nucleic acids. There's DNA and RNA. I give you a picture in your notes on page 13 of DNA and RNA. And basically, here's what you want to think. So I'm just going to draw a line here, and I know I'm chopping off some of this picture. But the RNA molecule, they're showing here, and then the DNA molecule is on the other side of my line. So I know it looks just like molecules everywhere, but they're giving us an example of each of those. Both DNA and RNA are made up of nucleotides. And so just as an amino acid, just as a protein is a chain of amino acids as the simple building block, a nucleic acid is also just a linear chain of a basic building block. That basic building block is called a nucleotide, and each nucleotide has three parts. There's a phosphate group, one phosphate, there's a sugar, which is ribose and RNA and deoxyribose and DNA, and then there's a base. Okay, we need to know that nucleic acids are made up of nucleotides, and we need to know the basic structure of a nucleotide. Okay, so I go through this in the notes. RNA is single-stranded. It's always single-stranded. But the thing is, the reason that there's different types of RNA is because this single strand, just like the protein, for instance, the single strand of amino acids can fold on itself, so can RNA. So with DNA, it's always a double-stranded structure. Those strands run in opposite directions, and you'll see arrows kind of denoting that. Or another simple way to see that is, do you see how the phosphate is here and here's the sugar? And so these are actually running in different directions. It's known as an anti-parallel arrangement. And then the bases project into the middle between the two strands. So here's the bases of one strand, and I can see how they bind to the bases of the neighboring strand. And that the bases bind together, and it forms kind of like the rungs of a ladder. If you want to think of it like a regular ladder, where your sugar-phosphate backbone makes the sides of the ladder, and then the rungs of the ladder are made out of the bases, which bind in a particular way, which is a purine, which is the double ring structure, will only bind with a pyrimidine, the single ring structure, or more specifically, C with G and A with T. So if we know the sequence of one strand of DNA, like here it's C, A, G, we can see this from the picture, then we can predict the sequence of the neighboring strand, or what's known as the complementary strand of DNA. So because we know C only binds with G, I can put a G there. Since I know A only binds with T, I can fill in T here. And G binds with C. And I'll definitely ask you about this on the test. So make sure that you understand those complementary base pairing rules. So I'm going to erase this. And just say that when you think about DNA, I would think about it as like the important document under glass. Nothing's going to interact with the DNA. Things interact with it, but like the DNA is not going to leave the nucleus and go out to the cytoplasm and interact with the organelles. But the RNA is really like the worker. The RNA is going to come in and out of the nucleus, and the RNA is really going to do the work as dictated by the DNA. So the question that I asked in our chat was, is the DNA in all of our cells identical? And you guys got that right. You know that it is identical. I'm just switching over to a picture here. And that's because all of our cells are the product of fertilization. So because, we originated as a sperm cell fertilizing an oocyte, 23 chromosomes came from the sperm, 23 came from the oocyte, giving us 46. That cell divided by mitosis and continues to divide by mitosis to give us every other, cell type in our body. Brain cells, muscle cells, liver cell, and so on. And so there is no other DNA that you could possibly have. All of our DNA is identical, and, great point about the criminal evidence that you made, too. So the way that I go about this in the notes is I look at something called the cell cycle. And so the cell cycle is depicted here, in our book. I'm just using my PowerPoint slides for a minute, where a cell's going to come into existence at one point. It's going to go through stages of its life in a certain order, just like us, just like infancy, childhood, adolescence, adulthood. Cells are going to go through stages of its life in a particular order, and certain developmental things happen during these stages. Okay, so the stages of a cell's life are G1, S, G2, and mitosis. So the way that I talk about DNA and RNA in the notes is I just go through the cell's life and I try to talk about what is happening with the DNA and the RNA, starting with the first phase of the cell's life, G1. What's happening there in G1 with the DNA? What's happening in S? And so that's how the notes read, and I'll show you that. So that's kind of like the story that I'm trying to build. So here's the thing about this picture in the cell cycle. Many, many times people have spent lots of time talking about like the replication of DNA that occurs in S phase, or the phases of mitosis, prophase, metaphase, anaphase, telophase. The thing is, these events are very short events. And so, let's say it's 24 hours for the DNA to replicate. G2 is usually like 2 hours, and it's under 24 hours for mitosis to occur. So my point here being that S phase, G2, and mitosis take, a total of under 3 days. So if a cell lives for 1 year, you're looking at 1 year minus 3 days that it's in G1. And so G1 is a very important period in the cell's life, because G1 is when the cell is doing the work that it's, meant to do. And that work is largely determined by the DNA. So since all of our cells have the same DNA, why would a white blood cell look different from a liver cell and behave different from a muscle cell, if it's the same DNA? And the reason that they look different and work different is because the genes that are being expressed by those cells are different. So in other words, think of the DNA as being the encyclopedia set of recipes on how to make individual proteins from A to Z. Okay, so it's the recipes of every protein we're ever going to need, A to Z, a gene being the recipe for a single protein. Okay, so that's what our DNA is. It's just this master recipe book on how to make every protein that we're ever going to need. And so in a liver cell, you're following the directions in the section liver. You have all the information, you're just not reading and following those directions, compared to, say, like a muscle cell is not paying attention to this liver page, but instead, is paying attention to all of the recipes contained in the muscle section, if that makes any sense. So that's just an analogy. So all of the DNA is there at all the cell types, but we're not expressing all of those genes in every cell type. We're expressing the genes that are supposed to be there that makes the cell look the way it's supposed to look and work the way it's supposed to work. And the way that it does that is through transcription and translation. So just to erase these notes, what's happening in G1 are these two processes, okay, called transcription and translation. So let's look at another picture for a minute. Okay, so in this picture, what they're showing us, and this is just a real simple depiction that the DNA, it's housed in the nucleus, that because the DNA contains all the instructions on how to make every protein you'd ever need, we need to deliver those instructions to the organelle that actually makes the protein. Now, the organelle that actually makes the protein is the ribosome. And so you can see here's our first major problem, because the DNA is located in the nucleus and the ribosomes are located out in the cytoplasm. So there's two separate physical locations. And because we said, you know, the DNA is going to be like the important document under glass, we don't want to really directly interact with it. We have to pass that on to our offspring, and so we want to keep that, you know, pristine, right? So what happens is when the DNA bubbles open within the nucleus, there are enzymes. They're called RNA polymerases. You don't have to worry about that detail, but there are enzymes that will copy that gene that's being exposed within the cell into a strand of RNA. So it will copy it into a strand of what's called messenger RNA. That messenger RNA would then physically leave the nucleus and go out to the ribosome. So do you see how the recipe that goes out to the ribosome, the organelle that actually makes the protein, is just going out into the cytoplasm? So this process of DNA going to messenger RNA, that process is called transcription. And then once the mRNA goes out into the cytoplasm, the way that the ribosome makes the protein, that's known as translation. So let me erase this and go to our notes. Okay, so here we are in the notes. So I talk about how the DNA, when it's in G1, exists in this form called chromatin. And I put a copy on the back of your notes of chromatin and chromosomes, how those differ. I made a video about that, so I'm going to leave it at that for now. But what I'm doing in the notes here, cell activities during G1, is I give you that analogy, and I'm trying to go through what happens, which is transcription. That's DNA being copied into that strand of RNA. And what happens to the RNA? Basically, it leaves the nucleus, it goes out to the ribosome, and then at the level of the ribosome, once the ribosome attaches to the mRNA, it's going to make the protein. That's what's called translation. And all of these notes are about translation. This picture, I love this picture. I use this in class. Here's your DNA double helix, bubbles open. This is when a gene is being exposed. An enzyme called RNA polymerase will automatically come into onto this open strand. And what it does is it takes individual RNA nucleotides and it builds a strand of RNA complementary to the DNA. That's all messenger RNA is. So here we're making a copy of that gene in the form of messenger RNA. And if you take a closer look at that, I can see that the RNA strand is complementary to the original DNA template, right? It's not identical, it's complementary, with, of course, RNA using the U instead of T in its structure. So once the messenger RNA goes out to the cytoplasm and goes to the organelle, the ribosome, what will happen is we're going to translate that into a protein. So the way that that works is that, and this is from your notes, that here's the ribosome, this bean-like looking thing. It binds to, this is your messenger RNA strand, and it reads at 3 bases at a time. Each 3-base sequence is known as a codon, and it's a code word for a particular amino acid. Unfortunately, the ribosome doesn't know what amino acid goes to which codon. You know, it doesn't know the genetic code, as it's called. Instead, all that happens is the ribosome holds technically 3 codons in place, and it waits for this other molecule called transfer RNA to bring the proper amino acid into the site. And the way we know it's the proper amino acid, the correct amino acid, is that there's a 3-base sequence on every tRNA that is known as the anticodon, which will try to base pair with the codon. And if it's complementary, it leaves that amino acid behind. So this tRNA is carrying a complementary anticodon to the codon. So let's erase that and look at the next picture. So what will happen is the ribosome is going to wait for another match. So now that we have the 1 codon match, do you see how here's a second tRNA coming in, and the anticodon is going to base pair to that codon. It's carrying an amino acid. So what happens is these amino acids bind together. The ribosome moves down the mRNA. That's going to eject that initial codon out of the ribosome. And what will happen is this tRNA is going to float away uncharged, or in other words, the amino acid was given to this growing chain of amino acids. So taking a look at this is what I mean. Codon ejected from the ribosome, transfer RNA floats away, what you call uncharged. There's the amino acid it was carrying, it donated it. But this tRNA is still bound to the ribosome. right, anticodon to codon. It's carrying the proper amino acid. But now we have a new codon in the reading frame, and it's waiting for another match. So here comes another tRNA, which you can imagine. I'm going to move down this picture. I was going to match and on and on. And so actually there are 64 different possible codons on messenger RNA, and 61 of them have matches. So eventually what's going to happen is you're going to get to one of these codons that doesn't have a match. You know, there's 3 of them. They're called stop codons. And at that point, all of this will fall apart. But you've made, you know, this messenger RNA is free. It could be translated again. And you've made the protein according to the instructions that came out of the nucleus. So I hope that little bit helps. If you need any other guidance on this, like any other parts of the notes, let me know, and I can make a video or answer any questions you have. I hope you have a great week. And what I would suggest is prepare for the test. Do the homework by the Wednesday night, of course. Take the test on Thursday or Friday, and don't worry about starting Unit 2, you know, until you're done with the test. I know I have Unit 2 out there, you know, up for grabs right now, but first finish Unit 1 to the best of your ability, then move on to Unit 2. Okay, talk again soon.